Hey, hi Regina.
Hello, Bo.
Hi.
Nice to see you.
It's nice to see you, and also it's nice to see we all have plants. I think you have you actually have a really nice garden. Yeah.
It's winter garden. I give all my lectures here. I like that environment for that.
Yeah, also, because I think talking about plants, also about other things, I think it's really nice that you have actually plants around you. So it's not just a bunch of humans talking about plants in abstract. Yes. Okay. I think it's really great to start today with you because you work on bacteria. The reason I started to think about how do plants practice politics was actually triggered by the term quorum sensing. So when I heard this term, I was really impressed. For people who don't know this term, it just means how bacteria can sense how many of them are in the same environment before they make some decision to do something, right? I was really impressed because the scientists decided to use a term that we humans use in political arena. So it's quorum, right? So quorum just means the minimum number of members of an assembly that must be present at any meeting in order to make the proceeding of that meeting valid. So I learned that the scientists, when they first discovered this phenomenon, that they didn't use this word. Later on, scientists decided to use this more political term. So that really triggered my thought, whether there are many things in nature, in other forms of life that we always tend not to use what we attribute to humans. So we tend to choose very different terms when we describe other beings versus when we describe what we do as humans. So that's actually the starting point of the inquiry. So it's great to actually talk to you about, actually we started with bacteria, even though this whole afternoon is more focusing on plants, but I think it's nice to start with bacteria because that's actually where my interest started. Yeah. You work on biofilm. Yes.
May I say something about quorum sensing?
Yes.
As you started with that, I actually know the scientist who coined that term. And as you said, initially it wasn't used. I mean, there were some other terms for it, but we even forgot about these initial terms. So at the very moment where this quorum sensing was used for the first time, I mean, there was some discussion among scientists whether we should use it because it comes from a different sphere, the political sphere. But once it was there, I mean, it was there to stay. And since then, it's used, and it's a very successful term. And it made actually quorum sensing, which is information exchange, communication between bacteria via chemicals, it made that very, very popular. So it really matters what kind of terms we choose in order to talk about science and scientific results, and also how we communicate that to the public. And for instance, also in lectures, what kind of terms we choose. So we often talk in metaphors, especially when we talk to students or to lay people, in order to actually get the message across. And it's becoming more and more popular also, even within science, to use these metaphors. It's a very interesting development.
Yeah, I was listening to Nature's podcast, Nature, the science magazine published in the UK. They just did a 3-episode series on some politics. I mean, just to make it a bit simpler for the time's sake, some scientists are quite reserved about using more political terms to describe what they study, and some, like you were saying, that are more comfortable. So, but maybe we can also get into this a bit later in the discussion. But before we get to these things, but I also want to start with science. So I had a great conversation with you last time in your lab, but I think many people may not know much about biofilm. Maybe we can start with a little bit of knowledge of biofilm. Yeah. So I'll hand it to you. Yeah.
Yeah. So biofilms are the multicellular form of bacterial life. So people usually think of bacteria as single cell organisms that just swim around in some liquid aqueous environment. But very often bacteria actually cluster together in big communities, and they're very dense. They grow together. They do everything together, basically. And what is really defining for a biofilm is that they stick together by producing an extracellular matrix, and this surrounds the cells, and it covers them, it protects them, and so they basically build a fortress. So a biofilm can be seen as a bacterial fortress. And this extracellular matrix consists of fibers, and there are different kinds of. So, and in the bacteria that we study, there are just two kinds of fibers, and one is a proteinaceous fiber, which is an amyloid protein. It actually has a similar structure as the Alzheimer plaques in our brain, very similar. And the other fiber is a form of cellulose. And these two fibers, they get entangled, and they are a very interesting material. They provide elasticity to these biofilms. And so they basically can grow very large. They can contain billions of cells, and they really behave like tissue. So they begin to fold and buckle up and have a beautiful morphology. And and basically why the bacteria are doing this is just they get protection, and they actually design their environment within a biofilm. So they build a cozy home, basically. Yeah.
So is the amount typical in billions, or it also varies in size?
It can vary in size. Sometimes there are just a few hundreds, there are a few thousands, but sometimes if you let them grow, as we do in the lab, there can be billions. And the biofilm can become so big that you can see it, and you can actually see this intricate morphology.
I thought also, talking to you and also reading the science papers you gave me, I thought it's very interesting because I think many of us do have this, I think including myself, before talking to you, we all have this imagination that bacteria are usually lonely swimmers in some environment, but you're saying they actually do build very large structures in their sense, right? So they build mega structures.
Yes. And they really cooperate in this building these structures. So there are different populations, subpopulations of the bacteria. Some build the matrix, others just grow and produce kind of pressure, so that basically the whole thing can buckle up and fold. So there's really division of labor. So it's basically a property of multicellular organisms already. So it's the beginning form of multicellularity, which is also one reason we study it. But another reason why we're studying is that these biofilms are very relevant in medical environments. So one property of these biofilms, or bacteria in these biofilms, is that they're highly tolerant against all kinds of toxic substances, like antibiotics, for instance. And so this means that if a biofilm is formed in our body, let's say in a wound or so, then the bacteria just hide away in these biofilms, and you cannot reach them by antibiotics anymore. And so this is the basis for chronic infection. And this is why biofilms are highly relevant medically. And this is why so many people around the world actually try to analyze and find out how biofilms work, and actually also how we can prevent them from forming or even dissolve them once they have formed.
So I have two questions. One is on the term biofilm, right? So I think I did ask you last time, because you in your description you also said castle or fort or megastructure, right? So how did this term biofilm come from? Because rather than calling them say megacities or megastructures, how did this term, and also it's not a very political term, right? Like what we were saying before. Yeah.
The term was originally introduced by engineers, because engineers also have a fight against biofilms, because biofilms build up in all kinds of tubes where you have some liquid. So it can be water, it can be petrol, whatever, on the surface of ships, for instance. And so it's an engineering problem to get rid of these biofilms. And there you have these surfaces, and as long as you don't look with a microscope, it looks very, very thin. And so it's a film on these surfaces, and it's of biological origin. So this is where the term biofilm originally came from. And like with other terms, it was popular and it stayed with us.
So do they all...
There are other forms of biofilms which are not films, I mean, like flocks or...
Yeah, I was going to ask, whether it's not just a flat structure, it can also be more three-dimensional.
Yes, definitely, yeah. Especially when you go to the microscale, when you look at it in a microscope, then you see the three-dimensional structure of it, which is very complex, yes.
And second question, because I think through whatever science we have learned over the past 100 years, whenever we hear bacteria or virus, it's always a negative connotation, right? So when I think even for me hearing the term biofilm because of association with bacteria, I have this negative feeling, right? And you also talk about its problem for engineers, it's also a problem for us, human health. But are there sort of, I mean, of course, for bacteria, it really helps them to survive, right? But outside the bacteria kingdom, are biofilms, can biofilms be also sort of more positive in some situations?
Definitely. I mean, that's just the general view that bacteria are harmful. And this has actually led us to use sanitizers, antibiotics, and just kill bacteria, which is a big mistake, because actually we live together with bacteria. We have them everywhere, on our skin, in our gut, everywhere. So basically, we are living ecosystems together with the bacteria. And And also the biofilms can be very helpful. For instance, in biotechnology, some things are produced in biofilms. In food technology, very classical food fermentation is very often happening in biofilms, like the production of vinegar, for instance, or things like nata de coco in Asian countries. These are all biofilms, basically. So and we eat them.
Okay. Yeah. Oh, so it's actually something we encounter every day. Yeah.
Yeah.
Okay.
Without seeing it.
Right. And last, do you want to show some images of the complexity of the structures that the bacteria actually build?
I try to.
Uh-huh.
Second. Let me see. Yes. So what you see here is the structure of a biofilm, and actually 3 different strains of E. coli. And the one on the right side, this is the so-called wild type. So it makes the 2 of these fibers that I mentioned earlier. So this is the curly fibers are the protein fibers, and the other one is this modified cellulose fiber. And this is the composite material that really surrounds the cells. So you barely can see the cells, basically. But the other 2 images, and this is false color, don't believe that color, is just to make it prettier. And you see in the middle, you see cellulose and how it flattens out and makes long-range connections. And on the left side, you see these curly fibers, the protein fibers, and they form kind of little baskets around the bacteria. And if the 2 of them come together, then we have what we see on the right side. And then it's really a very dense, elastic, very strong material surrounding the cells and really being very nicely protective.
So all the bacteria use these 2 types of fiber?
No. I mean, it's very common to have these amyloid fibers, but they're different kinds of. They don't have all the same protein sequence, but the same superstructure, the amyloid superstructure. And cellulose is also very common, but it's not there in all bacteria. I mean, there are other polysaccharides that bacteria produce in biofilms, but cellulose is quite common.
And last time you also showed me an image where they almost built a temple-like structure, right? So.
Yeah, this one?
Yeah.
Yeah, so this is how we also look at them under the fluorescence microscope. So we have certain dyes that actually stain this matrix, and so we give that into our petri dishes where these biofilms grow, and then we rapidly freeze the biofilm. So we cut them out from the petri dish, from the agar, then we freeze it, and then we cut it in very thin vertical slices. And then we put these or fix them, and then we look at them under the fluorescence microscope, because this matrix dye is a fluorescent compound, and this is what you see as green here. And so you can nicely see that this structure is very complicated. And so basically you can even see the cells, the black dots on the top here within this matrix. This is where the cells are, but the color is just the matrix. And you can see it's very dense here, and a little below you have these vertical structures that we call the pillars, and then you have some loose, more horizontal arrangement further to the bottom. And at the very bottom of these biofilms, we don't have any matrix. But there, the bacteria have flagella, and flagella are very long filaments, and they can rotate. And normally, if a bacterium is a single cell, then it uses this for being propelled, for swimming, basically, as a propeller. But here in the biofilm, these get entangled and also provide some cohesion in the biofilm. So you see, it's a very, very complex structure, and bacteria, billions of bacteria, are building this together coordinately, and it's a structure which is at least 100 times larger than the bacteria that build it. And I think this is absolutely amazing. And you can see it's like domes, and so it really has interesting forms.
Yeah, I mean, it would be really nice to do a 3D, sort of a 3D walk, just like in architectural model, to see that. Yeah.
Yeah.
How do these structures dissolve if they decide to go back to more sort of free swimming state?
Yeah, this is actually not yet really well studied. So I mean, this matrix, this extracellular matrix, is a lot of material. So it's carbon, it's nitrogen, it's all kinds of material. And in principle, one would expect that bacteria, when they don't need it anymore, they would actually take it in again and basically eat it up. We have not found that so far. So it's actually very difficult to dissolve. So this is why biofilms are very hard to eradicate. And we don't know how bacteria do that, how they get out, whether they really dissolve this again, or whether they just leave it somehow at the edges, or I don't know. We have not yet really studied that.
Do we know if they get out, at least?
I know that at the edges, so when this grows, as I can actually see to show you a colony. So this is what a whole colony biofilm looks like, and it's actually large. You can see it. It's 2 centimeters wide, and it's on a plate. And on the outer edges, this is the zone where the cells grow most rapidly. And at this position, if you just give them additional nutrient or water, a droplet of water, they can just swim out of it. So basically, yes, they can get out of it. Maybe not the ones which are somewhere in the middle, but at the edges, they can get out of it.
Okay.
And basically swim away, and then can start a new colony at some other place. Yeah.
I think I feel like maybe we should do another forum someday just about bacteria, because there are many questions to just think about the sociality of bacteria. But I'll move on to talk about plants for today. Are there more images to show, or?
Oh, maybe just one. One. And here we have the matrix actually in red. And these cells here, the very green ones, are the ones that grow very rapidly. So I said something about the two populations, those that grow rapidly and don't produce matrix, and in between you have the matrix producers. So this is just about this division of labor. And up here, basically all the cells grow slowly, so they don't light up in green, and then they produce the matrix. And here we have this very strong heterogeneity. So it's just a little bit more about the structure, but we can go to the plants now.
Yeah. But it's taking you how many years to get to this stage of research?
Oh, I have been starting to work on that some 25 years ago. And the focus on biofilms is, well, maybe I started 15 years ago.
Okay. But now I read, from the research paper, that you are also looking into how plants can help us humans to manage biofilm, right? So there's one paper I read, you sent it to me. It's about EGCG. It's a compound in green tea that can, does it prevent biofilm formation completely, or it kind of manages it?
First of all, there are no compounds or plant extracts that really inhibit biofilm formation of, let's say, all bacteria, something very general. It's usually specific, and they're very specific interaction between certain plants and certain bacteria. And because in nature, certain plants meet certain bacteria and vice versa. And so E. coli is a bacteria that we usually have in our guts. And so this means it gets out into the environment on a daily basis, basically. And so it gets into water, into soil, and of course on plants. And actually also birds have E. coli. So they just drop it and so it gets on plants. So plants in the environment get to see enteric bacteria, so gut bacteria like E. coli. And from the point of view of the bacterium, I mean, it would like to get back into a host, basically into the gut of a host, because this is a more cozy environment. You have constant temperature, you get something to eat, and well, you know what you have around you, basically. But in the environment, life is much tougher for bacteria. So everything can change all the time. You have radiation, you have nothing to eat, you can have high salt, low salt, whatever can happen. And there's no control about that. So bacteria would like to get back into the gut environment. And the easiest way to do that is basically to just stick to plants and hope, basically, that you get eaten again by some animals or even humans. And they actually use this type of biofilm to stick to plants. And now, what does that mean for the plant? Now, the plant has these bacteria on its surface, and like all macroorganisms, so plants, animals, we have huge surfaces which are nicely colonizable by bacteria. So that means, okay, we cannot avoid bacteria, but we have to control what kind of bacteria actually colonizes there, so that there are no harmful bacteria, so that we get in a good relationship with these bacteria. And the plant has the same problem, basically. So the plant wants to control what kind of bacteria and in what state they are on its surfaces, basically. And biofilm, making thick biofilms, is also not good for the plant, because in this biofilm, the bacteria kind of hide away, as I said before, and they cannot be reached by communication molecules, basically. Many of these molecules, like, they can be bound by the matrix, and then they don't reach the cells, and so on. So basically, the plant has an interest not to let the bacteria grow too thick biofilms. So as long as it attaches to the surface and is not harmful, it's fine. Well, actually, some bacteria can become harmful for the plant. They actually can poke holes into plant cells and start to feed on the plant cells, which is not good for the plant. This would be a pathogenic bacteria. But E. coli usually doesn't do that. Yeah. But the plant wants to control it. And so the control involves some sort of communication, and communication by plants is usually chemically. And so the plant produces chemicals that kind of control biofilm formation. And at some point, I mean, I said before, so we have a strong interest finding compounds or agents that inhibit biofilm formation, because we want to use that for medical purposes. And so at some point, I thought that, well, we should look for that in plants, and specifically in plants that have been traditionally used in order or in therapy of chronic infectious diseases, like chronically infected wounds, because there we have the biofilms. Right. And so these plants that are useful for this purpose should have very powerful anti-biofilm compounds. And this is how we started to work on plants and the interaction with the bacteria, and thinking about which plants we could actually choose. And that turned out to be very successful. And in parallel, we also look at the chemicals that these plants contain. So for many, especially medicinal plants, it's very well studied what they contain, actually. So there are long lists of compounds, and many of them can be just bought. So we buy them and look whether the compounds have the same effects as the complex plant extracts. And sometimes we find the active compounds this way.
So just one question in my mind as I was listening to this, because you were saying earlier that we actually don't know whether the bacteria can get out of the biofilm once they build it, right? So let's say if plants actually encourage E. coli to build biofilm, then when we eat the plant, the bacteria cannot get out. But if they discourage the bacteria to form biofilm when we eat the plant, So I'm not talking about medicinal plant. I'm just talking about, say, other maybe more major grains, like cereal, right? Like rice or wheat. So this is just, I mean, something come up to my mind. I mean, I don't know if this is relevant. Yeah.
Well, I think in general, the plants tend to inhibit biofilm formation on its surface, not to promote, unless it's a bacterium which is useful for the plants. And there are, of course, examples like the bacteria in the root nodules that fix nitrogen for the plants. And so the establishment of these bacteria in the root nodules also involves biofilm formation. And there the plant actually actively invites the bacteria. So it attracts them and tells them, yes, make the right biofilm at the right position and fix nitrogen for me. And they get food for it, basically. So this is a symbiosis, a win-win situation.
Yeah, no, I'm just thinking if plants actually encourage them to build biofilm, if they cannot get out of the biofilm, then when we eat it, then it's not a problem for us, right? So it's also, there's another way maybe to sort of lock them in biofilm in the environment, so they cannot swim in our guts. I mean, this is just a random, I mean, this is just crazy.
But I think the plants have not evolved to protect humans. And so this is actually something that very few people think about. We use all kinds of compounds in traditional medicine from plants against all kinds of diseases. And you never think, why should a plant make a certain compound that helps, I don't know, against some kind of human disease? I mean, kind of a chronic infection by a bacteria, there is the link, because this bacteria might also be attacking the plants. So there is a rationale behind it. Yeah, so the plants to help us, basically. But there is, of course, an environment, well, in our gut, in our intestine, where we eat plants. And so plant material and the bacteria, our endogenous microbiome, basically directly interact there. And if we eat plants that, for instance, contain these antibiofilm compounds, especially wild plants, not bred plants that have been bred for a large yield. They usually don't make those anymore, but the wild plants really make them. And then this is actually good for us because especially E. coli and related bacteria that makes these amyloid fibers, this is not good for us because these amyloid fibers promote inflammation.
Right.
So if we eat...
So it's not just the bacteria, it's also these fibers that make us sick.
Yes. I mean, at least it contributes. It has a pro-inflammatory activity, these fibers. And so if we can inhibit the formation of these fibers in our intestine with these plant compounds, it's actually good for us. And that's one of the many reasons why, for instance, flavonoids in plants are healthy, because they reduce a tendency to inflammation. They reduce this, and so by many processes. But one of the processes is by suppressing these fibers, which are made by bacteria and which are pro-inflammatory.
So, just specifically to biofilm, why do plants have these chemicals that can suppress biofilm formation? Do we also have some chemicals that we can produce on our own that can suppress biofilm formation?
Yeah, I mean, first of all, the plants, why do they have them? Because they having thick biofilms on their surfaces would result in the bacteria being no longer reachable by communication molecules. So they want to suppress this or reduce the biofilm formation. What was the
We want to, right? But I'm just saying, why evolutionarily so far, is it the case that we have less sort of anti-biofilm chemicals that we can produce?
We actually have different ones. So we have, for instance, antimicrobial peptides, which are produced in our gut. And these are usually seen as, in high concentration, they are toxic for bacteria, but in a somewhat lower concentration, they can actually inhibit biofilm formation. So we basically use the same strategy. We produce small molecules, but a different kind of peptides to suppress biofilm formation.
So we have some, and then plants have their own, right? So now we're trying to learn from to understand what the plants can produce.
Yeah.
Right.
So the plants usually use some sort of polyphenols. So there are myriads of polyphenols. They're catechins, they're flavonoids, tannins, etc., etc. And so these are plant-specific compounds, and we produce different ones, but the strategy is the same.
I see. And so the article I read is about EGCG, this chemical compound from green tea. But now you have looked at not just green tea, right? So you've also looked at other plants. How many plants have you identified that have some biofilm suppression compound?
We actually have quite a number. It's still a small number, but because we also look into the mechanism, how they actually act. And this is a lot of work. So we have to focus on some plants which are really promising and really have strong and interesting activities. But the plants we were looking for came from two different origins. So we were looking in medicinal plants, traditional phytotherapy, and one set of plants comes from Cretan, from the island of Crete. So where traditional medicine is still very much alive, so there are still people who know a lot about that, and it is basically a tradition that dates back from antiquity and even earlier into Minoan times.
Yeah.
And so I can get these plants and even extracts prepared the way the people do it there, because I have connections there. And the other origin of plants we use is from Ayurvedic medicine from India. And because a few years ago I had a postdoc from India, and she knew something about Ayurvedic medicine. And also Ayurvedic medicine is now commonly used also here, and you can buy the products in a standardized form. And so with both kinds of plants from Crete and from Indian plants, we have found quite a number of plant extracts that strongly reduce biofilm formation. And now we're looking into the details. What kind of compounds are active, and how do they do it?
So why not look into local herbal tradition?
Because yeah, we could in principle also do this, but it's well, yeah, in principle, yes, we could do it. But I just use these connections to Crete because I can get all this. And because the tradition is still very much alive, so we can collect this information about how to use these plants and against what kind of diseases and so on. And it's very well documented, and also for Ayurvedic medicine. And there are actually lots of scientists also working on Ayurvedic medicine and how they act in the human body. So there's a huge body of literature that we can actually use.
And I was really taken when you said last time that you use this word negotiate, right? So you said the plants actually negotiate with bacteria, so they can manage their relationship, right? So what do you mean by negotiation, rather than sort of a just I'll kill you, that kind of scenario?
Yeah, exactly. So the most simple interaction is mutual killing. And this is what bacteria usually do when they compete for food, basically. Imagine you have two populations of bacteria. There's a limited amount of food they're eating, they're growing, and then the food is basically exhausted. And during this transition into that exhausted state, bacteria many of them can actually produce some toxin, and they just kill the other population in order to get everything. This is very simple and straightforward. But the relationship between plants and bacteria, and humans and bacteria, and humans and plants are much more complicated. So this involves communication. And because there are different interests, and there has to be, you have to compromise, and if everything is fine, then it's a win-win situation. And in evolution, actually, it usually starts with some negative interactions. So what we now see with the coronavirus, it bumps into the human population, and it's a disaster. But over time, the two actors, basically, they get used to each other, they adapt to each other. And at the end of a long evolutionary time, there can be symbiosis because they have adapted so well to each other. And so all this involves lots of complicated interactions and communication processes. And this is what I call negotiation. So it's that both sides have certain interests, and they try to find a way that somehow they get along the best way. Yeah.
So there are two questions. We can dive a bit deeper into this. I think this is sort of the core. So there are two processes. One is time, right? So you said, usually it takes a long evolutionary time for symbiosis to be negotiated. So I know you are a molecular biologist, right? So do you also look into the evolutionary history of bacteria and biofilm and plants, or you need to collaborate with someone to do the evolutionary study?
Well, we don't really look into these evolutionary aspects, except for we using sequences of proteins and genes, and of course you can derive insight into the evolution from comparing different sequences. Right. And E. coli, by the way, our main bacteria, is very interesting also from an evolutionary aspect, because it is hugely diversified. It has many different strains that can do very different things. Also have different kinds of biofilms and so on. And so we can actually look at the genes. So we have all this genome information, compare the genes and the proteins and see how they diverge from each other and what that might mean. Right. And so we actually do some of this work also by actually comparing harmless E. coli that we all have in our gut with pathogenic E. coli, which from time to time emerge, like EHEC or enteroaggregative E. coli and so on.
The second question is when in our human imagination, when we talk about negotiation, it's a very verbal, it's a very language-based process, right? So so when you say negotiation, do you mean that there's also sort of molecular signaling happening, or it's some other forms of negotiation?
Yeah, it's definitely molecular signaling. So we have molecules. It's chemical communication basically, and especially plants. I mean, unlike animals, animals can also in their interaction they can run away, they can bite if they're attacked, but the plants cannot do that. So whatever the plants do, I mean, they can have thorns, okay, but it's mostly chemical. So plants are very good in producing all kinds of communication chemicals, of toxic chemicals if they get attacked, and so on. And of course, they use chemicals to communicate with bacteria. And bacteria and plants also have certain receptors, so they can sense each other and sense these kind of chemicals. And they have signaling pathways that then trigger certain reactions in both of them, basically. And this is how they communicate and interact, basically, and organize their coexistence.
Right. I'm thinking, I don't know if this is right, I'm just thinking, in our way of life, often of course, some languages are performative, meaning like, by speaking, by enunciating something, that's already an action. But often we separate language from action, right? So we will say something and then we do something, right? So from the plant and bacteria perspective, the molecular is both signal but also action, right? Or there are different signals that belong to different categories. You understand what I'm asking, right?
Yeah. In principle, it's hard to make this separation because I mean, you have the production of a communication chemical. It's being sent out, it's diffusing around, it reaches the other partner, and it's being sensed. I mean, it's a whole sequence of events, and yeah. And some of these chemicals are very specific. Now, actually. Since you said whether there is some kind of more general language or more specific language, actually,
I meant that. So no, I think I didn't say it clearly. I meant this. So I meant when, let's say, the plant release some compound, and then the bacteria can receive it. So but the compound is not only something like our language, but it's also like our action. It actually inhibits certain action of the bacteria. So the molecule serves as both saying and doing at the same time. Because we tend to say something and then do something, and these two can be separate in most situations.
Okay. Yeah, I mean, there actually these kind of molecules different sorts of molecules. So, for instance, if the plant sends out a flavonoid or this EGCG in green tea, then these molecules can directly affect the assembly of these fibers. So
Right.
it's a direct chemical effect. And
Right.
it does not have to enter the bacterium in order to do that. But
Right.
on top of that, some of these compounds are also poor signaling compounds, because there is a receptor on the surface of the bacteria that is bound by this compound. And then the rest is an information transfer. It's no longer a material transfer, but it's a signal transduction pathway into the cell, which then might actually switch off or on certain genes, produce new proteins, and so trigger a reaction, basically, and a new behavior of the bacterium. For instance, we have found that one of the plants, one of the Ayurvedic plants, actually triggers the bacteria to become better swimmers.
Right.
And so this is clearly a signaling molecule that affects some sort of signal transduction pathway in the bacteria that we still haven't figured out. We don't know what it is. But what happens is that the bacteria make more of these flagella, the motility organelles, and they start to become more motile. They swim around. And so basically the plant tells the bacterium, I don't want you on my surface, swim away. And it forces it to produce the swimming organelle, so it actually leaves. Yeah.
So do you think it, I mean, like, there are two ways, I'm thinking, right? So one is it useful for biologists to distinguish, say, language versus action, language versus material? You know, one compound actually stops them from doing things, and one compound is kind of like a linguistic order for them to do something, right? I guess for you, it's not necessary to put them into different categories.
Yeah, because it's always material. At least some step in the sequence is material. There are molecules involved, and molecules that can bind, that can combine with something or trigger then some additional pathway. And so some steps are usually material, and others are then information transfer, but it's also material. So we can actually look into the details of what the molecules do to each other in a whole sequence of processes, basically. So we don't have to distinguish something like a pure language or so. Yeah.
Right. I mean, of course, I think the mirror side, the effect is also interesting. I think it also perhaps can prompt us to think about our understanding of language, right? Of course, people talk about performativity, but whether we can even think further to see language are really material. I mean, I haven't thought about this. This could be interesting for us.
I mean, it's sound waves, and sound waves are also kind of material.
Yes. Yeah. So in terms of negotiation, when you said it's more in a win-win situation, so the bacteria can still survive. They just don't do biofilms to completely destroy the plant. So do you think this is how plants manage its microbiota in general, or it's there are different strategies they use. It's not just all negotiation.
Yeah, but it's communication, and as I said, every microorganism with large surfaces has to do this, because I mean, you cannot clear bacteria from your surfaces. It's too difficult, and then you actually also open the space for the bad bacteria, for the pathogens, basically. So the strategy has to be, well, get the right bacteria on your surface, and then you are protected, because they actually occupy the space, and so the pathogens have a harder time to enter and also colonize. And of course, this involves this kind of negotiation and communication, chemical communication, to attract the right bacteria and make sure that not the wrong ones are coming and settling down.
Right, because I'm thinking in terms of, when we think about sort of managing a complex situation, it's not just a one-to-one relationship, right? It's not just the EGCG and then the E. coli. It's also managing other relations with other bacteria. So there are many complex sort of negotiations going on to achieve some kind of balance, right? Do you think this is also the situation?
Oh yeah, definitely. Because I mean, if you look at the whole system, it's usually much, much more complex. So in science, we of course we focus on specific processes, that, especially in molecular biology, because we want to understand these details. But if you look at the whole system, there are so many processes going on in parallel, and so we're basically looking at complex ecosystems. If you look at what happens in our gut, we have all this plant material, everything we eat basically. We have hundreds of species of bacteria. They all negotiate and communicate basically. And if this goes well, then it's nicely balanced and it's to our benefit as well. If it doesn't go well, then we have the wrong bacteria or bacteria at the wrong ratios, basically, proportions, and then our microbiome is in disbalance, basically, and which can make us sick and has severe consequences. And all these relationships are countless and very complex. Yeah.
So you're kind of saying, even with one plant negotiating with its microbiota, there's kind of like what's going on in our human society, right? So there are many negotiations going on, but it's just more on a molecular level that we don't see or notice at all.
Yeah, definitely. Yeah. And also on surface, there are so many different bacteria and insects and so on. There are many players. And you have to take into account all of them, which is very difficult. And they have very interesting strategies to actually, well, interact, help each other, attack each other. Others help you against the attacks from some others. And
Right.
especially plants have very interesting strategies to, for instance, recruit, let's say, some insect to combat other insects that have attacking them, for instance, by laying eggs on them or so. Yeah.
Right. I was thinking whether they also can recruit other bacteria to manage a sort of bacteria that they don't like.
In principle, that's the general approach. You let the good bacteria grow basically on or have them on your surface, so they occupy the space, and the bad ones cannot enter the space as easily. Yeah, and then you also bacteria plants have these. Well, the very helpful bacteria, like the ones that fix nitrogen and which are actively invited to actually really grow at certain positions in the plants where they should be and where they can help the plant. Mm-hmm. And this is root nodules or so. Yeah.
Yeah, I mean, last time you showed me the pathways for the E. coli signaling pathways. I mean, it took you 15, 20 years to understand a complete picture. I mean, for me, that's so incredible. But now I'm thinking, for us to understand how plants actually manage the microbiota on its surface, I mean, that's going to take, I don't know how many centuries of human research to really understand, right?
I don't know about centuries, but it will take time. Yes, but it's interesting. It has to be done, and it will take years. Yeah, and I also think we will never be able to understand all the details. So what we do in the lab, we choose some interesting processes which can serve as a model system. So we want to find out how certain basic processes in these model systems work, and we know then we have similar proteins, genes, etc., in other bacteria or in other living beings, and we can extrapolate from that. So we don't have the illusion that we can understand everything. Right. In long times we won't be able to do that. Yeah.
Let's talk a little bit about Latour, Bruno Latour, French sociologist, philosopher, because you said he's one of your heroes, right? I don't think I hear this often from a scientist. So which of his ideas were in particular that you find heroic for you?
I think recently Bruno Latour has turned a lot towards biology. And in one of his last books, he actually develops the strategy that we actually have to reintegrate ourselves into the ecosystems on this planet, in the critical zone on the surface of these planets where everything happens, basically all life processes. And I think we definitely also as scientists we should do that. We should understand that we are part of all this, and that we are not outside, basically just looking at that from outer space, basically, yeah, and that we are actually part of that, and not only scientists, but we as a society as a whole, because I mean humans have traditionally looked at that as, well, the planet is resources that we can use, and in the process of using, we convert it into waste. So resource to waste, and it's a linear process. And by that, we are turning the planet into waste, basically. And this cannot go on. So we basically have to reintegrate into these circular processes where there is no resource and no waste. It's just transformation. And I think science is very important in order to understand these processes and guide us how we can actually do that. And I think this is also more or less what Bruno Latour says in much nicer words, of course, and coming from philosophy and philosophy and biology actually meet.
I'm curious, you know, I'm asking, I think this is out of curiosity, but also I think it's very relevant. Like, how do you think more scientists can be more open to sort of inquiries like Latour's, to really think about the culture nature divide, whether this divide should be maintained or not, right? How, you know, I'm very grateful to be able to talk to scientists here, but I feel in like kind of going back to the first, when we started talking about quorum sensing, using a term that has a use in human politics as well. How do you think, for you in particular, why are you more open-minded to not to keep the culture nature divide? Like, what helped you to move away from that sort of objectivity paradigm?
I think because of my personal history, I actually come from a family of artists and designers, and that has always drawn me towards that field as well. And so now at the Humboldt-Universität, I have these collaborations with designers, cultural theoreticians, etc., people from the humanities, architects, material scientists, and so on. And so this very strong interdisciplinary work. And but I think I have been open to that because of my background and breadth of interests, basically. But I think in general, this is rarely found in scientists, and this has a very simple reason, because science means if you work in the lab, you work every day from the morning to the night. You really have to focus on something. It's a lot of work. It requires a lot of your time, basically. And so many people just don't have the time to read other things, And so it's a small minority of scientists who actually are interested in these aspects, but they exist. And you find them in these interdisciplinary cooperations, basically.
Yes, I mean, I've been very fortunate to talk to you, to talk to Rusa, to talk to Matthias. So I think there are scientists who are open to crazy conversations. Yeah.
There's actually one other thing that just comes to my mind. Right. So I have always looked at as humans as since the very beginning when I studied biology, and I actually also had some professors that gave lectures that went this direction. That humans are, I mean, they are a product of evolution. We are animals. We're smart animals, but we are a product of evolution. And so the attitude that here are the humans and here's the rest of nature, and a human dominates that, and this is different categories, has always seemed to me kind of unlogic. Because we belong basically to it, so we part of this system, yeah, and so that just comes naturally to me, also as a biologist and thinking of humans as biological beings, basically.
I think we only had like 15 minutes to talk about plants and biofilm and bacteria, but I think just a very short conversation. I hope it's so clear that plants are not as stupid or simple as or even bacteria are not as stupid, as simple as we used to think. So I think I feel I think many of us want to move towards more sort of humble direction. I think science definitely helps me. And I think it's just whether the research can be integrated also into sort of philosophical worldview that help us to change. Yeah. So thanks so much. And then we'll talk more at 4 p.m. And now we'll take a break. The weather's really nice. So next session starts at 2:30. We will go, I and Dr. Rusa Leiden, we'll go out and talk on the phone while we're walking. And I hope and many people, I hope many people who are listening can also just join us at 2:30. Just listening, but also to walk in nature. I mean, the weather is so nice today. Yeah, and then we'll be back at inside at 4 p.m. Okay. Yeah, thanks, Regina. We'll talk again at 4. Yeah.
Thank you, and see you again.
See you.
For bye-bye.
See you at 4. Yeah.